Welcome to Evolutionary Biology. Today, we are going to explore one of the most fascinating concepts in nature, heritable variations. We will discover exactly how traits are passed down from one generation to the next and why these tiny genetic differences are the absolute fundamental fuel for all of evolution. Let us dive in. To understand what heritable variations are, imagine you have a secret family recipe for chocolate chip cookies. If your grandmother accidentally writes down 2 cups of sugar instead of 1, every future generation will bake sweeter cookies. This is exactly how heritable variations work in nature. Instead of a cookbook, the instructions are written in DNA. When a living creature acquires a change in its DNA, like a thicker fur coat, and passes that exact DNA to its offspring, that trait becomes a heritable variation. It is a change in the biological recipe that allows a family line to slowly change over time. Now that we understand the basic idea through our recipe analogy, let us look at the formal biological definition. In evolutionary biology, heritable variations are the genetic differences among individuals in a population that can be transmitted from parents to their offspring. But where do these differences come from? They primarily arise from 3 sources: random genetic mutations, genetic recombination during sexual reproduction, and gene flow between different populations. These mechanisms constantly shuffle and alter the genetic deck, ensuring that no 2 individuals are exactly alike, which is crucial for the survival of any species. While genetic differences are passed down, we must carefully distinguish them from environmental variations. Environmental variations are changes caused purely by an organism's surroundings or lifestyle. Think of a human getting a sunburn, or a tree bending because of strong coastal winds. Because these environmental changes do not alter the underlying DNA sequence in the reproductive cells, specifically the sperm or egg cells, they simply cannot be passed on to the next generation. Therefore, only variations that are physically encoded in the genome qualify as heritable and can contribute to the long-term evolutionary process. So, why do these heritable variations matter so much? They are the absolute fundamental raw material for evolution and natural selection. Imagine a population of identical clones facing a sudden deadly virus. Because they all share the exact same genetic weaknesses, the entire population could be wiped out in days. However, if there is heritable variation, a few individuals might possess a mutation making them naturally immune. They survive, reproduce, and save the species. Similarly, human farmers rely heavily on these genetic differences to selectively breed crops for more food, proving their essential role in both nature and civilization. Now that we know why these variations matter, let us look at how biologists measure them using a mathematical concept called phenotypic variance. This formula tells us that the total physical differences we see in a group of organisms are simply the sum of their genetic differences and their environmental differences. VP represents the total phenotypic variance, which is the overall physical variation we can actually measure. VG stands for the genetic variance, caused strictly by differences in DNA. Finally, VE represents the environmental variance, caused by outside factors like diet or sunlight. If the environmental variance increases because organisms live in wildly different habitats, the total observable variance will also increase, even if their genetics remain exactly the same. Once we know how much of the variation is genetic, we can calculate a crucial value called broad sense heritability. Conceptually, this formula is asking what fraction of all the physical differences we see is strictly due to DNA. The symbol h squared represents broad sense heritability, which is always a ratio between 0 and 1. We calculate it by dividing the genetic variance, Vg, by the total phenotypic variance, Vp. If h squared is close to 1, it means almost all the variation is genetic, and the trait will respond very strongly to natural selection. If it is close to 0, the environment is causing almost all the differences, meaning the trait cannot easily evolve. Let us see this formula in action with a concrete example. A botanist is studying wild sunflowers and finds the total phenotypic variance in height is 120 square centimeters. Through experiments, she determines the environmental variance is 30 square centimeters. We are asked to find the genetic variance and the broad sense heritability. First, we rearrange our variance equation to isolate the genetic variance, subtracting 30 from 120 to get 90. Next, we plug this into our heritability formula, dividing 90 by 120. The final answer is 0.75. Physically, this means 75% of the variation in sunflower height is due to heritable genetic differences, meaning the trait can easily evolve. Now that we have calculated heritability, what can we actually do with it? Heritability acts as a powerful predictive tool. It shows us exactly how strongly a population will respond to selection pressures over time. If a farmer knows that a trait like plant height or milk production is highly heritable, they can selectively breed only the best individuals, confident that the next generation will inherit those superior traits. This brings us to a mathematical way to predict the exact future traits of a population, using something called the breeder's equation. This brings us to a mathematical way to predict evolution, starting with the selection differential. Conceptually, this measures how much better the chosen parents are compared to the general population. In this formula, S represents the selection differential. The symbol μS is the average trait value of the elite breeding parents, and μO is the average trait value of the entire original herd. By subtracting the original average from the selected average, we quantify the exact strength of the selection pressure being applied. If a farmer chooses only the very largest animals to breed, the selection differential will increase, representing a strong push to change the population. Once we have the selection differential, we use the famous breeder's equation to find the response to selection. This formula beautifully shows the evolutionary response depends on both how hard you push the population and how much of the trait is actually controlled by genetics. The symbol R stands for the response to selection, which is the actual evolutionary change we expect to see in the offspring generation. We calculate it by multiplying the heritability, h squared, by the selection differential, S. If heritability is low, even a massive selection differential will result in a very small response because the traits are simply not being passed down effectively. Let us apply the breeder's equation to a real-world farming scenario. A dairy farmer has a herd producing an average of 20 liters of milk per day. He selects the best cows, averaging 25 liters, to breed. The heritability is 0.4. We need to find the new generation's average milk production. First, we find the selection differential by subtracting 20 from 25, giving us 5 liters. Next, we multiply this 5 liters by the heritability of 0.4 to get a response to selection of 2.0 liters. Finally, we add this 2-liter improvement to the original 20-liter average to find μN. The final answer is 22 liters.
Physically, this proves the offspring improve, but do not perfectly match their parents, because the trait is only partially heritable.
To visually reinforce what we just calculated, let us look at a parent-offspring regression plot. On the horizontal axis, we have the average trait value of the parents, known as the mid-parent value. On the vertical axis, we have the trait value of their offspring. Each dot represents one family. Notice the clear positive trend. Taller parents tend to have taller offspring. The solid line running through the data is the line of best fit. The most important feature of this diagram is the slope of this line. In evolutionary biology, the slope of this exact regression line is mathematically equal to the heritability of the trait. A steeper slope means higher heritability and a stronger genetic link. We have covered a lot of ground today, so let us review the key takeaways. First, heritable variations are the essential genetic differences passed from one generation to the next. Without them, populations would be completely static and unable to adapt. Second, we learned that total physical differences, or phenotypic variants, are a combination of both genetic and environmental factors. Finally, by calculating heritability and using the breeder's equation, we can predict exactly how a species will evolve over time. You now understand the mathematical and biological foundation of how every magnificent adaptation in nature begins. Excellent work!